Athlete DNA secrets. What if the difference between a gold medal and a career-ending injury wasn't just training, dedication, or luck? What if it was a switch? A single biological switch buried deep within the double helix of an athlete's DNA, waiting for the right signal to either unlock godlike potential or slam the door on a promising career forever. This isn't the plot of a sci-fi thriller. It's the terrifying theory whispered in the most elite, high-stakes corners of the sporting world. They call it the Lazarus Gene, a forbidden sequence, a genetic failsafe, or more chillingly, a kill switch that could represent the final, unconquered frontier of human performance. To understand this earth-shattering concept, we have to move beyond the simple idea of good genes. We already know about specific genetic markers that give athletes an edge. Think of the ACTN3 gene, often called the sprint gene. Certain variants produce a protein, alpha-actinin 3, found exclusively in fast-twitch muscle fibers, giving sprinters and power athletes an explosive advantage. Or consider the Finnish skier Eero Mantaranta, who had a natural genetic mutation in his EPO receptor gene, allowing his body to produce an abnormally high number of red blood cells. He had, in essence, a natural, undetectable form of blood doping, which let him dominate his sport in the 1960s. These are fascinating, but they are static advantages. They are part of the baseline genetic lottery. The Lazarus gene is something else entirely. The theory posits a dynamic genetic element, a gene or a cluster of genes that doesn't just grant a passive benefit, but can be actively regulated, turned up, turned down, switched on, or switched off. Imagine a gene that controls the efficiency of mitochondrial energy production. Most of the time, it operates at a normal, baseline level. But what if a specific biological trigger, perhaps an extreme physiological stress, a unique nutritional signal, or even a synthetic molecule could flip its switch? Suddenly, an athlete's cells are producing ATP, the body's energy currency, at a rate 10 or 20% higher than their rivals. Their endurance becomes limitless. Their recovery time, once measured in days, is now measured in hours. This isn't just an enhancement, it's a complete paradigm shift in human biology. Now let's consider the darker side of this switch, the kill function. This is where the theory becomes truly unsettling. The same mechanism that could grant superhuman abilities could also be used to revoke them. Perhaps this gene also regulates the process of apoptosis, or programmed cell death. Flipping the switch in the opposite direction could initiate a cascade of cellular breakdown in key muscle tissues. An athlete at their absolute peak could suddenly experience rapid muscle wasting, chronic fatigue, or an inability to recover from even minor exertion. Their career wouldn't just decline, it would be terminated at the molecular level. It would look like a mysterious, untreatable illness, a tragic case of burnout, but its origins would be encoded, a secret hidden within their own cells. How could such a mechanism even exist? The answer may lie in the field of epigenetics. Epigenetics is the study of how behaviors and environment can cause changes that affect the way your genes work. Unlike genetic mutations, epigenetic changes are reversible and do not change your DNA sequence, but they can change how your body reads a DNA sequence. Think of your DNA as a massive library. The genes are the individual recipes. Epigenetics determines which recipes are read and which are ignored. Mechanisms like DNA methylation and histone modification act as bookmarks and sticky notes, telling your cellular machinery to read this recipe or skip this chapter. The Lazarus gene theory proposes that there could be a critical athletic gene with a highly sensitive epigenetic marker. A specific targeted intervention, a designer drug, a specific frequency of electrical stimulation, a tailored protein, could be developed to attach to that marker and change its instruction. It could remove a stop sign that was suppressing the gene, unleashing its full potential. Conversely, it could add a stop sign, effectively silencing a gene responsible for crucial functions like muscle repair or oxygen transport. The athlete wouldn't be genetically modified in the traditional sense. Their hardware would be the same, but the software running on that hardware would have been hacked. This makes it almost impossible to detect with current anti-doping methods, which primarily look for foreign substances or known genetic mutations, not the subtle epigenetic signals that control a person's own native DNA. This is the ghost in the machine, the ultimate biological exploit, a power that could make champions or break them with terrifying precision.
Genetic control sports. The theoretical science is one thing, a fascinating and frightening puzzle for geneticists to ponder. But what happens when this theory escapes the lab? What if it's already out there, a clandestine tool in the arsenal of a rogue Mason or a shadowy private syndicate obsessed with victory at any cost? This is where we move from the world of biology to the ethical abyss of genetic control in sports. The pursuit of the Lazarus gene represents the final and perhaps most dangerous chapter in the long and sordid history of performance enhancement. We've gone from crude steroids in the 70s and 80s to sophisticated blood doping with EPO in the 90s. Now, we stand on the precipice of gene doping, an invisible, intrinsic method of cheating that rewrites the very definition of a natural athlete. The World Anti-Doping Agency, or WADA, has been worried about this for years. They've already banned gene doping, defining it as the non-therapeutic use of cells, genes, genetic elements, or of the modulation of gene expression, having the capacity to improve athletic performance. But how do you police something that leaves no trace? If an athlete's own body is producing the performance-enhancing substance because its genetic instructions have been subtly altered, what do you test for? You're not looking for a needle in a haystack. You're looking for a single piece of hay that's been instructed to behave like a needle. It's a regulatory nightmare. Now, let's inject the kill switch concept into this reality. The implications are chilling and go far beyond simple cheating. It becomes a mechanism of ultimate control. Imagine a state-sponsored athletic program that identifies promising young athletes with the latent Lazarus gene. They are nurtured, trained, and at the opportune moment, a secret protocol is used to activate their potential. They shatter world records. They bring glory to their nation. They become national heroes. But they are living on borrowed time, their abilities leased to them by their handlers. What happens if this athlete considers defecting, or speaks out against the regime, or simply gets too old and is no longer useful? A simple, targeted intervention could flip the switch. Their powers would fade, their bodies would betray them. The world would see a tragic story of an athlete who burned too bright, too fast. No one would ever suspect the truth. The athlete becomes a biological asset, one that can be activated, managed, and ultimately decommissioned. This isn't just about fairness in sport anymore. It's about fundamental human rights and bodily autonomy. Would athletes even be told what was happening to them? They might be given a vitamin supplement or a recovery therapy without ever knowing its true purpose. They would feel invincible, like they'd finally unlocked their true potential through hard work, never realizing they were a pawn in a much larger genetic chess game. This technology would create a new, invisible class system in sports, the genetically activated and the natural. The entire concept of a level playing field, the very spirit of the Olympics, would be rendered meaningless. It would be a competition not between athletes, but between the bioengineers and geneticists working in the shadows behind them. The potential for abuse is staggering. A corporation could patent the activation sequence for a Lazarus gene and sell it to the highest bidder, creating super teams that are literally unbeatable. A criminal organization could use the kill switch as the ultimate form of blackmail against a star player, forcing them to throw a game under threat of having their career molecularly dismantled. The very idea turns athletes from inspirational figures of human achievement into puppets, their strings pulled by unseen masters who hold the keys to their biological code. This is the terrifying future that the pursuit of the Lazarus gene could unlock. A world where human potential is not discovered, but assigned, not earned, but activated, and can be cruelly and silently taken away. It forces us to ask a terrifying question. In the relentless pursuit of breaking human limits, are we on the verge of breaking humanity itself? What do you think is hiding in the DNA of our greatest champions?